The planet Earth supports a variety of life forms. Millions of different species of organisms. Microorganisms are one such variety. The word microorganism or microbe is used for an organism that is so small that it cannot be seen without the aid of a microscope. Microbes are ubiquitous in nature. They are found everywhere if, for a short time, we are provided with an electron microscopic vision. We would see these microbes, millions and billions of them everywhere. On our hands, beneath our nails, on our teeth, on our table surface, on pillows, on the tip of a pin, the most pervasive ones. Bacteria and cyanobacteria are prokaryotes. Prokaryotic cells lack the true nucleus. Their genetic material is not surrounded by a nuclear membrane. Fungi, protozoa, and algae are the eukaryotic microbes. They have a true nucleus enclosed by a nuclear membrane. The advanced forms of microbes are usually multicellular. Most of the fungi and algae belong to this category, producing filament-like structures. Many of these microorganisms are known to us because they are associated with disease and misery. But however much we may dread these tiny things as the causative agents of contagious disease, a great majority of microorganisms are essential to all life forms on Earth. They decompose the complex biomolecules from dead organisms and organic wastes. They are found in association with plants in a symbiotic relationship, and they help maintain the fertility of the soil. Microorganisms also synthesize a large number of valuable industrial products like the microbial fermentation process also provides a number of useful products. Examples are for these functions, microbes have been exploited to aid production at commercial levels. They also aid in the bioremediation process and act as biocontrol agent. They have been used as model organisms for the investigation of genetical and biochemical processes. They are much easier to work with compared to plants and animals. Millions of copies of a single cell can be produced in a short time and at a low cost to give plenty of homogeneous experimental material. This encouraged the scientists to exploit them as tools in the field of biotechnology, the recombinant DNA technology.
The ability to obtain the microorganisms in order to study their characteristics depends on being able to culture on the growth medium in the laboratory conditions. A microbial culture consists of innumerable number of cells that originate from single cells or few cells and spores. These cells are cultured on a nutrient medium under very specific conditions, both of the nutrient as well as the physical factors. One important aspect of microbial culturing is to maintain aseptic conditions. This is a multi-step process that consists of sterilization of the media, the implements, and the other conditions that are going to be used for raising the culture.
For sterilization, all the glassware is properly closed, packed in suitable containers, or wrapped in aluminium foil. Glass containers like flasks and test tubes are closed either by caps or cotton plugs. To make a cotton plug, a small piece of muslin cloth is spread on the mouth of the container. The required amount of cotton wool is stuffed to close the mouth completely. The muslin cloth is then tied around the cotton. Heat-resistant glassware and implements are sterilized by dry heating in an oven above 120 degrees Celsius for 16 to 18 hours, that is, overnight. The heat-sensitive material and media are sterilized by steam under pressure by the autoclave. The autoclave is an instrument that works on the principle of a pressure cooker. Glassware can also be sterilized by autoclaving. The autoclave is provided with a water tank where water boils with the help of an electric element. The steam so generated comes in contact with the material placed in the chamber. The steam is allowed to build till the temperature reaches 120 degrees Celsius and the pressure achieved is 15 pounds per square inch. Sterilization under these conditions for 15 minutes is effective in killing all microorganisms and heat-resistant spores. After 15 minutes, the instrument is switched off and allowed to cool till the pressure comes to 0. The lid is then opened and the material taken out. There are certain precautions that one should take. Water in the tank should not be less or more than the specified amount. The lid of the chamber should be closed properly. Proper pressure should be allowed to be built. The instrument should not be forced open unless it has cooled down and the pressure comes to 0. In case one doesn't have access to an autoclave, a pressure cooker can be used. Once the pressure reaches, heating should be allowed to go on for 30 minutes. Certain components of the media like amino acid, vitamin, blood sera, and other organic compounds are destroyed at the temperature of autoclaving. These materials are made in solution and filter sterilized. The filters used are essentially membrane filters. Their pores are so small that bacterial cells and spores cannot pass through them. So, a solution passed through these filters becomes microbe-free. The work area for carrying out microbial work should also be sterile. The most common practice is to use the laminar flow hood for such type of work. The hood is a partially contained workspace. Inside the chamber wall, high-efficiency particulate air filters or HEPA filters are placed. The air that passes through these filters becomes sterile. The larger sized particles in the air are filtered by the pre-filters. So, the air coming from outside as well as the air escaping from the work area is decontaminated. Horizontal laminar flow has HEPA filters on the front wall of its hood, and the vertical laminar flow has HEPA filters on the roof of its hood. An additional facility for sterilization in this hood is ultraviolet or UV light, which must be switched on for at least 30 minutes before starting the work. UV rays are lethal to microbes. It is a must to switch off the UV while working, as these rays can adversely affect both human skin and eyes. The airflow and light of the hood must be switched on before starting the work. The working platform is cleaned with a disinfectant or rectified spirit before and after every use. The flame from the spirit lamp or a Bunsen burner heats surrounding air and sterilizes it. In the absence of the hood, microbial work can be carried out on a clean workbench using burners or spirit lamps. Microbes are not different from ourselves. They require a combination of appropriate nutrients and physical conditions to grow. A mixture of nutrients is the culture medium on which the microbes are grown in the laboratory. When the constituents of the medium and the chemical composition of these constituents are known, the medium is a synthetic medium. It is called the defined medium. Non-synthetic media are complex nutrient media that are of organic origin. Though their exact constituents are not known, they can support the growth of a variety of microbes and are useful for routine laboratory culture. The medium can be used either in a liquid form or jellified with agar-agar to provide solid support. In order to grow the microbes in vitro, the first step would be to prepare the medium. The medium we are going to use here is the peptone dextrose medium. Weigh 10 grams of peptone. And 40 grams of dextrose. Dissolve these in 100 milliliter distilled water in a 1 liter flask. Make the volume up to 900 milliliter with distilled water. Set the pH in the range of 6.2 to 6.5. Either with 1 normal hydrochloric acid or 1 normal sodium hydroxide as required. Now, make the volume up to 1 liter with distilled water. Dispense the medium into 2 flasks, pouring 500 milliliter into each. Weigh 7.5 grams agar-agar and add in 1 flask to solidify the medium. This will make 1.5% by weight the concentration of agar. For liquid medium, the required volume is dispensed in the flask or tubes. Measure 40 milliliter of liquid medium in a 100 milliliter measuring cylinder and pour into a 250 milliliter flask. If the volume is small, say 10 milliliters, the medium can be dispensed with the help of a dispenser. Dispense the medium in the required number of flasks. Plug all the flasks. Cover with aluminium foil and autoclave. Pouring and dispensing of the medium should be done inside the laminar flow. The solid medium at the time of pouring should be in a molten state. Take a sterilized test tube. Pour 10 milliliter of medium in this tube. Put this tube in the test tube rack. You can dispense the medium in the required number of test tubes in the same manner. Tilt the rack in such a way that the medium takes the shape of a slant. Leave the tubes in that position to solidify the slants. Discard the used pipettes in the disinfectant. Dilute potassium dichromate is the disinfectant used here. Take a test tube and fill it with the required volume of water. Mark the tube at the level of water. Empty the tube. Now use this tube as a graduated tube. Hold the empty sterilized tube alongside this graduated tube. Make the corresponding marking on the sterilized tube. The empty sterilized tube is now a graduated tube. Take out the sterilized Petri plates from the can. Take a sterilized test tube, graduated up to 30 milliliters. Pour the medium in this tube up to the mark. Open the lid of the Petri plates partially. Pour the entire medium into the plate. Secure the lid. Prepare the requisite number of plates. Leave the plates to solidify the medium. It takes almost an hour to solidify. Small volumes of liquid medium can also be dispensed after autoclaving. Discard the used pipettes. The liquid and solid media are now ready. The cultures can be transferred onto them. In the next part of the program, we will learn the procedures of culture transfer and growth and disposal of studied microbial cultures.